Good morning, everyone. My name is Dr. Brian Booth from the Department of Bioengineering at Clemson University. And this morning, the title of my talk is Redirection of HER2-positive breast cancer cells results in phenotypic and genotypic changes. So I should start with, what do we mean when we say redirection of cancer cells? Well, I think a picture will help demonstrate what we're talking about. Basically, redirection of cancer cells is when we can force tumorigenic cells isolated from mammary tumors or human breast cancer cell lines to lose their tumor-forming capacity in vivo, and we've also developed an in vitro system that mimics this. So as you can see here in these tumors, these are mouse mammary tumors that formed from a transgenic mammary tumor that overexpresses the LAC-Z gene. So we use the LAC-Z gene in this case as a marker of cancer cells. It's easily identified by this blue stain. So when LAC-Z interacts with XGAL, you get this nice blue stain. So we isolated cells from these mammary tumors, grew them up in culture, transplanted them into new mice, and we have mammary tumors formed. So this is what you'd expect for normal tumor development. In redirected cells, what happens is we take these same cancer cells and mix them in specific ratios with normal mammary or breast epithelial cells. And when we transplant these into new recipient mice, so these are juvenile mice, female mice, pre-pregnancy, all the endogenous mammary epithelium has been removed, and we transplant these cells into the remaining mammary fat pad, and what we see is normal development. So you see the development of normal mammary ducts. We have side branching, some tertiary branching here, and these are comprised of blue cells. Now, the only blue cells in these transplants come from cancer cells. So you see this blue stain. All of these mammary ducts have cancer cells that have been incorporated into them. Now, depending on the ratio, we can completely attenuate the tumor-forming potential of these cancer cells. So if you look at a cross-section, you notice that these ducts are not comprised entirely of blue cancer cells. There are non-blue cells indicating the presence of normal mammary epithelial cells. So it's a mixture of cells that are transplanted, and you see this mixture of cells in these normal outgrowths. So those are virgin mice, and when we get these mice pregnant, they differentiate and form milk-producing cells again. So in this case, we took human cancer cells, specifically human testicular cells. That's a whole nother story that you can read about here in Boussard et al. But these human testicular cancer cells formed normal mammary outgrowths, and during pregnancy differentiated into milk-producing cells. So we use specific antibodies, and we stained for mouse caseins. You see it here in red. And in this case, human alpha-lactobumin, showing that the presence of human milk in these lactating mammary glands. So what I want to talk to you now is what we've been working on for the past couple months, or years, I guess. So like I said, we developed an in vitro model that mimics the results that we see in vivo. So in this case, you see here, these SKBR3 cells were transfected to constitutively express red fluorescent protein. Seen here, this is just a 2D in vitro culture. So we've done this with multiple human breast cancer cell lines. We use HER2-positive breast cancer cell lines because previously I found that the HER2 on these cancer cells is overexpressed in tumors, and in the redirected cells, the receptor is still being expressed, but the activation has been attenuated. So the phosphorylation is gone. So we use HER2 as a biomarker. So the continued expression of HER2 allows us to sort these cells out of culture or out of in vivo outgrowths. So if you take these RFP cancer cells alone, transplant them into mice, you get nice mammary tumors that are full of RFP positive cells, indicating that they come from these cells. When we mixed these HER2 positive cells and normal epithelial cells in a 1-to-1 ratio, grew them up in co-cultures, magnetically sorted them, transplanted them again. You see some normal outgrowth in this mammary gland, but you also see a very nice, or well, it's not nice, but a RFP positive tumor that is formed. When we co-culture them in a 1:50 ratio, so this is 1 cancer cell for every 50 epithelial cells in culture, grow them for 4 to 6 days, and then transplant them again, you see a lot of normal mammary development, growth, epithelial development. On higher power, you can see that these cancer cells, these RFP cancer cells, have been incorporated into these new regenerated mammary ducts. So what we've done recently is we've sorted these cells out and done some RNA-seq here. So these are the cancer cells here. You see specific patterns of gene expression here. So these are the cancer cells. Over here, these are the normal cells. And for each population of assorted cells, we did the RNA-seq. So right here, these 3 columns are the redirected cancer cells whose gene expression patterns match the normal epithelial cells. If we compare the cancer cells to the in this case, the MCF10A, which we use as our normal breast epithelial controls. In this volcano plot you see here, so in these plots, the dashed blue line, or the blue line, this is the adjusted p-value threshold. So anything above this has a p-value of 0.05, and then the red lines here are full change thresholds. So either it's a 2-fold increase or 2-fold decrease, depending on the comparison. So you see thousands of genes that are differentially expressed between the cancer cells and the normal epithelial cells. And there's HER2. And also CD44, which is a marker of breast cancer stem cells, and TGF-beta1, which is a popular gene in cancer research. So we did the comparisons for all of our sorted populations. And in this case here, in G, this is our 1:50 HER2 negative fraction compared to our epithelial cells. So you would expect both the very little genes that are differentially expressed, which is what we'd expect, because these are basically epithelial cells being compared to epithelial cells in slightly different culture conditions. We move down to H. These are the normal epithelial cells RNA-seq patterns compared to the redirected cells. Very few genes that are continue to be differentially expressed. So again, these redirected cells are moving from a cancer genotype or gene expression patterns to a normal gene expression pattern. And if we look here, this is our redirected cells compared to the cancer cells. Normally, you would expect them to look like this, but if you look at these ones, this looks almost identical to the cancer cells compared to the normal epithelial cells, but these are the cancer cells compared to the redirected cells. And we further did some bioinformatic intracellular signal pathway investigation. So you see here, these are the top pathways that are differentially regulated between the cancer cells and the normal cells. And this graph here, or this table here, are the pathways that are differentially expressed between the cancer cells and the redirected cells. Anything in bold appears on both lists. So there's some interesting and suspected targets or pathways here: IL-6, the JAK-STAT signaling pathway, and then you have your EMT, epithelial mesenchymal transition, P53. All of these pathways are many times differentially regulated during tumor development and cancer progression. So in conclusion, these are tumor cells are redirected, which occurs both in vitro and in vivo. Our redirected cells differentially express genes compared to normal and tumor cells, and redirected cancer cells move toward a normal genetic profile. And I just want to acknowledge people who have done this here at Clemson. Dr. Frank Kamenetsky did a lot of the recent research. She just graduated. Caroline Jordan has taken up the project now. She's working on it. Dr. Feltis and Kim Roche, well, she's now a doctor, they did some of the initial bioinformatics work. And then at the National Cancer Institute, Dr. Smith and Thomas Meyer did the majority of the bioinformatics works that I just presented. So with that, I thank you for your attention, and I will, I have to cite our funding sources, INBRE, of course, and the Clemson Creative Inquiry. And with that, I will be happy to try to answer any of your questions. Thank you.